Thank you, Andy, for that very kind introduction. I think I'm giving the same talk you heard at that European flight meet. Not sure much has happened since then. Look, it's a wonderful privilege and honor to give one of the Max Birnstiel lectures. I knew Max, not well, but I met him a few times when I was a postdoc in Walter Gehring's lab back in Basel, Switzerland in the 1980s. And there's actually, I mean, he seems like a very staid and sober guy, you know, very Swiss. But there's actually a few very funny stories about him. And so I just thought, you know, so I went through all the stories, and I think this is the best one. The time he created an international diplomatic incident. So, you know, all these Swiss guys take their military service in the Swiss Army very seriously. And it's like a militia. They have to go every year for 2 weeks until they're 43 years old. So it's not just one tour of duty for a year or so when they're young. It goes on forever. Max was very proud of his military service. Many scientists are just privates, you know, they're just, you know, at the lowest edges of the hierarchy. But Max became a lieutenant, and he was in charge of a small artillery unit that specialized in detonating mortars. You know, so they, you know, these little cannons, these little mortar deployers, and they would send these salvos of 5 or 10 shots of mortars at a time. So his unit was in the east of Switzerland, and they were doing their exercises. And then Max, you know, yelled, "Fire," right? And the mortars were pointed the wrong way. They were supposed to be pointed to the west in this open sort of practice field in Switzerland, but instead they were pointed east. And these 5 or 10 shells went into the tiny country of Liechtenstein and blew up. And it blew up a barn. No one got hurt, fortunately. It blew up a barn, and it really did create some diplomatic tensions between Switzerland and Liechtenstein. Never even heard of Liechtenstein. But anyway, they were freaked out. They thought Switzerland was invading them. So this was all. Now, Max apparently was very proud of that story when he was outside of Switzerland. So he would go abroad. He would brag about how he invaded Liechtenstein. But when he was in Switzerland and in the company of fellow Swiss soldiers, he never talked about it. Okay, I thought that was pretty good. Okay. So Max is best known for a pioneer in isolating eukaryotic genes. And he isolated some of the very first genes, the ribosomal genes. He later went on to isolate the histone genes and actually identified kind of a precursor of enhancers, which is today's topic, called the modulator element, which regulates the levels of histone expression. Among others, Max trained the great Walter Schaffner, who was the discoverer and namer of enhancers. Walter was a postdoc with Max, and then Max kept Walter on as an assistant professor in the Molecular Biology Institute that he founded at the University of Zurich before coming here to Vienna to start the IMP. And it was shortly after Max recruited Walter Schaffner to the University of Zurich that Schaffner in his lab in 1981 discovered the transcriptional enhancer. So I think Max would approve, at least in principle, not much of me, but at least the topic of enhancers. Okay, so that's what I'm going to talk about today. I think there's intellectually a direct link between what I'm going to talk about today and Max's passion to understand gene regulation, which is really what he devoted most of his scientific career to. Now, you know from Alex's work here and others that enhancers are short segments of genomic DNA. They're typically a few hundred base pairs in length, and they switch genes on and off in response to a variety of cellular signals. The human genome is thought to contain something like 400,000 enhancers. That's a lot of enhancers. That averages out to something like 10 to 20 enhancers per gene in our genomes. It's a lot of regulation. And there's mounting evidence that sequence polymorphisms in enhancer DNAs are a major source for population diversity and predilection to disease. My lab has used the Drosophila embryo as a sort of assay system to study the core properties of enhancer DNAs for a very long time. And most of our studies focus on a 1-hour interval between 2 and 3 hours after fertilization, when approximately 1,000 enhancers regulate the expression of several hundred patterning genes, which are expressed in a series of stripes and bands to establish the blueprint of the adult fly. And so that's what I'm going to focus on today. And an example of this process is shown on this first slide. And Andy already mentioned this. So this is an old slide. And it shows the expression of a segmentation gene called even skipped, or Eve. You see that in those 7 brown stripes. This is a 2.5-hour embryo, fixed and then stained with antibodies to show the Eve expression pattern. These stripes foreshadow the subdivision of the embryo into a repeating series of body segments over the next several hours during embryogenesis. And we found that these stripes are regulated by a series of separate enhancers located both upstream and downstream of the Eve transcription unit. All of these enhancers show a surprisingly similar principle of gene control, and most of our efforts focused on really nailing down one of those enhancers, the one that controls the expression of stripe number 2. And over several years of studies, using these kind of static snapshots, these fixed tissues and antibody staining, gave us a lot of insights on the spatial limits of gene expression, how localized gap repressors, for example, form the borders of Eve stripe 2. But these studies gave very little information about temporal dynamics. Why does this stripe form exactly 30 minutes into the cell cycle, quite reproducibly in embryo after embryo? So to get at temporal dynamics, as opposed to spatial dynamics, we've turned to live imaging methods. And I got to tell you something about these live imaging methods. I had a long-term plan that Andy alluded to. About 10 years ago, I made a decision that I was going to switch from flies to sea squirts and devote the lab to evo-devo studies on evolutionary origins. Of course, I used to joke that if some old guy is talking about evolution, it's time to stick a fork in him. He's probably done. Okay, so I don't know what this says about me. So I feel a little like Michael Corleone in Godfather 3. Every time I try to get away from the fly embryo, it keeps pulling me back, pulling me back. I cannot leave it. And so most of my lab does sea squirts now, but what happened that pulled me back was the live imaging. So I'm going to show you a movie of the stripe 2 expression pattern in a living embryo. Visualization of the nascent RNAs depends on MS2 stem loops. These are about 50 nucleotide loop structures in the nascent RNA, which can be visualized very, very efficiently and sensitively with a bacteriophage protein called MCP attached to GFP. This method was developed by Rob Singer at Einstein University and then adapted for use in fly ovaries and embryos by Liz Gavis and Thomas Greger. So we're basically using their method. You're going to see green dots in individual nuclei. Those are sites of active de novo transcription. Those are transcription foci, and we can visualize 3 or 4 RNAs. We're probably right at single cell detection, but we really don't push it too hard. We're happy to visualize 3 or 4 transcripts or more at any given time. So this is a high magnification view of a transgenic embryo that has the stripe 2 enhancer attached to a reporter gene with the MS2 stem loops. You'll see the transcription foci at the periphery. They're shut off at mitosis. They come back on. Now we're entering this 1-hour period I mentioned where the blueprint is established. Localized repressors form the borders of Eve stripe 2, and no sooner do you get the mature stripe that it disappears. Okay, so this is very beautiful. When I saw this, I thought, no, I can't just work on sea squirts. We got to continue to work a little bit on flies. So we're currently looking at different mechanisms for timing and gene expression, looking at those things that we just couldn't study using fixed tissue preparations. And I'm just going to summarize 2 mechanisms for the control of temporal dynamics, strictly timing. One is summarizing. I'm just going to summarize these studies so I can get to the main event of enhancer promoter communication and transcriptional bursting. One mechanism of timing, in the case of Eve stripe 2, depends on enhancer-enhancer interactions within the complex Eve regulatory region. And so this is all summarized here. We know that the Eve locus is organized in a topological associating domain, or a TAD, by virtue of these interactions between flanking insulators N-HOMI and HOMI. And this loop domain is very real. You can see it through high-C assays, and it was identified by Paul Shadle and Jim Janes at Thomas Jefferson University. This shows the different stripe enhancers in the 5' and 3' position relative to the Eve transcription unit. And I don't want you guys to despair back there. I only have something like 18 slides, all right? I don't have that many slides. So we're going to get through this. Your brains will be intact. I promise you, maybe you won't learn anything, but your brains will not release random information as I speak. It's just too short, not too many slides. So we found that when we take the stripe 2 enhancer out of this locus and attach it to a reporter, as I showed you in the previous slide, it comes on way too early. And the dynamics are delayed. It doesn't refine the way the endogenous stripe 2 does. And that's because of inhibitory interactions between the stripe 1 and the stripe 2 enhancer. If we look at the endogenous Eve locus and monitor stripe 2 expression, it comes on prematurely and it's delayed in its refinement if we remove the stripe 1 enhancer. If the stripe 1 enhancer is intact, on the other hand, stripe 2 comes on at the right time, comes on late, and it sharpens up with the correct dynamics. And so we believe that repressors bound to the stripe 1 enhancer in this position of the embryo, which keeps stripe 1 off, attenuate and modulate the timing of stripe 2 expression. And we see this over and over again. A lot of evidence for enhancer-enhancer interactions. Inactive enhancers are not dead. Many of them have repressors which will modulate the activities of other enhancers in the locus. So I'm just going to summarize that point. A second mechanism of timing that we're currently studying, and this is done by a postdoc in the lab, João Raimundo, is timing in the looping of distal enhancers to the target promoter. So here you're looking at cartoons showing the expression of a gene called brinker, which is expressed in this lateral stripe in the early embryo. And normally, brinker comes on at nuclear cycle 13. That's the one cell division cycle prior to this 1 hour period when the Eve stripes form and the body plan of the adult is established. Now, the enhancer that controls this pattern is located at 11 kilobases upstream of the brinker transcription start site. And in this location, we know who the activators are. When the activators bind to this enhancer, they're not enough to switch on gene expression. There's a delay. If we take that same enhancer, or when João takes that same enhancer and he puts it close to the promoter, within a few kilobases of the brinker promoter, it comes on prematurely, it comes on early. In other words, when the enhancer is close by, as soon as the activators bind to the enhancer, the gene is switched on. But when the enhancer, in this case, is in its normal distal location at 11 kilobases away, the binding of the activators are not enough. There's another tier of temporal gene control, and that's the looping of the enhancer to the promoter, as summarized on the next slide. It's a very fancy slide made by João. So the idea is, you've got this distal enhancer. It's Drosophila N of kappa B, which is present very early in the embryo, is the primary activator, which binds to these sites. And that binding is not enough to switch on expression, because this enhancer is far away. It's 11 kb away. João noticed that many of the genes active in the early fly embryo that have distal enhancers more than 5 kilobases away depend on GAGA binding motifs. So there's GAGA elements, GAGA, either within the enhancer or next to the enhancer, and there's GAGA elements in or next to the target promoter. And we know that this GAGA binding factor, the GAGA associated factor, binds to these GAGA elements to mediate looping, which you see here. And this loop brings the enhancer near the promoter. As I will argue, it doesn't touch the promoter. That's going to be a major point of my talk, is that enhancers do not and cannot touch their target promoters. They come into proximity of about 3 or 4 kilobases. Once they come in within a few kilobases, now the gene is finally turned on. So this temporal delay is due strictly to the GAF binding protein, how long it takes to bind these GAGA elements and mediate the looping to bring the enhancer into proximity. This all occurs within a topological associating domain. Victor Corsi's lab and others have shown that the Drosophila genome is organized in a series of loop domains through insulators that are distributed every 30 to 50 kilobases throughout the Drosophila genome. Okay, so these are two mechanisms of temporal control that we really couldn't study using the old-fashioned fixed tissue preparations, enhancer-enhancer interactions, and the regulation of looping interactions. The most obvious thing that came out of our studies using live imaging is transcriptional bursting. Every gene we've looked at in the fly embryo shows bursting. So even when a gene is fully on, it flickers on and off. And I'm going to show you an example for the segmentation gene. Whoops, I'll go back. For the segmentation gene Fujichuatsu, or FUTS, that's like even skipped. It's expressed in a series of 7 stripes along the length of the embryo. And the visualization was done by inserting these MS2 RNA stem loops into the endogenous FUTS locus in the 3' UTR. If you put these stem loops up in the 5' UTR, it inactivates the gene because you don't get good translation of the FUTS mRNAs. But as long as it's in the 3' UTR, this is totally non-invasive. This allele works perfectly well, and homozygous flies, where both of their copies are tagged with MS2, work just fine. You know, we have stocks of flies that seem perfectly happy as homozygotes with the stem loops. And so we're monitoring the de novo transcription of FUTS using these 3' MS2 stem loops, again, visualizing with the MCP bacteriophage protein attached to GFP. And what I want you to notice is in the 30-minute time window, when the 7 FUTS stripes form along the length of the embryo, there's a lot of flickering of the signal in individual nuclei. And those are transcriptional bursts. You look at any nucleus, and it's flickering on and off. And a typical burst runs for 3 or 4 minutes. During this time, we believe that something like 20 RNA polymerases are recruited to the promoter. They're released once every 10 seconds or so. So over the course of a couple of minutes, you release this train of polymerases, 10 or 20 polymerases. They elongate along the DNA template to produce a burst, then they fall off, and then you get another burst. And how long that period, that refractory period of inactivity occurs depends on the enhancer, and I'll come back to that point in a moment. Now, it turns out that in early embryos, prior to this nuclear cycle 14, when the blueprint is established, at the earlier stages, there are very rapid nuclear divisions, and the interphase only lasts for 5 to 7 minutes. And that's only time basically for one burst. So sometimes you'll squeeze out 2 bursts, but typically an interphase nucleus at these earlier stages show just a single burst. And some of the genes active at very high levels in the early embryo, and there are not that many, even skipped is one of them, show as part of the bursting process transport of all the burst products of the 10 or 20 RNAs made from a single burst are transported on the mitotic spindle. And these are new results from João Raimundo, and I'll show one example. So this is a high magnification view of a single nucleus at an earlier stage during a period of very rapid mitoses. Here's the single burst that occurred during the 5 to 7 minute interphase, and this nucleus is getting ready to undergo mitosis. And what you'll see is that this little burst of product, there's 10 or 20 transcripts there, are packaged as a unit, as a granule. They'll jump onto the spindle, and then they'll get transported to one of the daughter nuclei. So you'll see the chromosomes will condense, the packet is released, and it moves along the spindle ahead of the segregating chromosomes into the daughter nucleus. And this is more easily seen in the still shots. This is a sad fact. You know, we make these beautiful movies, and then we analyze them by just looking at stills because the movies are just a little overwhelming. So this is just a series of still shots that I showed you in that previous movie. There's the burst. As the chromosomes condense, all the transcripts of that single burst are released and then move on the spindle. So we see this transport process as a continuation of the bursting event that occurred in the previous nuclear cleavage cycle. And João believes that this is a mechanism to ensure equal transport of these very precious transcripts made in the early embryo. When you only get a single burst, you're only making about 10 or 20 products at a time, that there's equal transport of this. And some evidence for this view that this is a purposeful transport process to ensure that the daughter nuclei get equivalent amounts of product from the earlier burst is shown here. This embryo is a heterozygote. It just has one tag copy of the endogenous even skip gene. This one is a homozygote where both copies are tagged. And you see the bursts from homologs at opposite ends of the nucleus. Here the chromosomes are condensing. You can see the nascent transcripts, or the RMP, are popping off their respective homologs, attaching to the opposite spindles, and then moving in an orderly fashion to their respective poles. So it's just, these are early days, and it's preliminary data. And I don't know why we missed this after 35 years of doing fixed tissue in situ hybridizations, but we missed this. And as Joao looks, he sees that maybe half the genes expressed in the early embryo show this kind of mitotic transport process. And again, we see this as part of the bursting process. So there's not just a transcription burst as an inherent property of gene expression, but there is apparently processing and transport of the products of that burst as a unit. All right. So what's the relationship? I'm going to spend the rest of the time discussing the relationship between enhancers and bursting and what this tells us about enhancer-promoter communication. And I am specifically obsessed with one question. How close does an enhancer have to get to its promoter in order to trigger transcription? Now, you may see this as a modest question, but I have a little bit of obsessive-compulsive personality disorder, and I got it into my head that we have to know this distance. Okay, so for those of you who want to take a nap, right, the answer is 100 to 200 nanometers. Okay, but let me show you how we got there. All right. So first off, the relationship between enhancers and bursting. So this shows the expression, or summarizes the expression of two different reporter genes containing MS2 stem loops at the 5' UTR, and then two different enhancers, both from a gene called snail, placed 3', 8 kilobases downstream of the transcription start site. The snail shadow enhancer, which is a distal enhancer, it's a fairly strong enhancer, and the weaker proximal enhancer. And we know from qPCR assays that the shadow enhancer, the distal enhancer, produces twice as many transcripts in the early embryo as the proximal enhancer. It also produces twice as many transcriptional bursts. So what you're looking at here are quantitative traces from single nuclei in living embryos that express each of these two reporter genes. The x-axis is time in minutes, so we're looking at about a 45-minute time window. The y-axis are arbitrary units of fluorescence intensity, which scales with the number of elongating polymerases and the number of nascent transcripts that are made. And the main difference between this strong enhancer and this weak enhancer are the number of bursts. So the snail shadow enhancer, which produces twice as many transcripts as the proximal enhancer, produces about twice as many bursts as the weaker proximal enhancer. But the burst size are very similar between these two enhancers. The burst size is about 3 to 4 minutes and is producing something like 10 to 20 RNAs. The main difference is the stronger enhancer produces more bursts, but the burst size are about the same. And we see this as a consistent trend. Stronger enhancers produce more product because they generate more bursts, not bigger bursts, but more bursts over a particular period of time. Now, when we first got these results several years ago, we thought that the bursts were due to unstable enhancer-promoter interactions. Our thinking, my thinking, was completely dominated by the classical model put forward by Walter Schaffner and others, that distal enhancers loop directly to the promoter, and that maybe that's an unstable interaction. So maybe these bursts come from instability. So in other words, the enhancer, a remote enhancer, loops to the promoter, lingers for a few minutes, produces a burst, and then falls off. And then stronger enhancers like the snail shadow enhancer are able to re-loop to the promoter more quickly than the weaker proximal enhancer. Seemed like a very good model. It's completely wrong. And I'll show you some evidence that it's wrong. So to test this idea, Takashi Fukaya and Bomi Lim placed a number of well-defined developmental enhancers, weak and strong, in this central location, downstream, as I've already mentioned, of this MS2 reporter gene, but also downstream of a second reporter gene that contains PP7 RNA stem loops. This is an orthogonal bacteriophage system. These transcripts could be detected with the bacteriophage protein PCP attached to either M-cherry or tandem tomato. So the nascent transcripts made from the reporter gene on the left are visualized in green, those on the right in red. And we really did expect to see, and the experiment was established to see alternating red and green bursts. We thought, okay, this is a completely symmetric system. The enhancer is 8 kilobases downstream of both promoters. We're using the same minimal snail promoter sequences in both locations. So the enhancer would just randomly select one of the promoters, produce a burst, fall off, and then once again it would be jump ball, and the enhancer would select one of the promoters. And the net effect would be alternating red and green bursts. But that's not what was observed. What we have consistently observed was coordinated bursting of the reporter genes. So this shows a quantitative trace of a single nucleus from a living embryo that expresses this two-color reporter system. The x-axis is time in minutes, so we're looking at a period of about 30 minutes. Once again, the y-axis is fluorescence intensity. And you can see, far from getting alternating red and green bursts, we see fairly coordinate bursts. In this case, just 3 bursts were produced in the 30-minute time window of the analysis. And these and other findings like it, I believe, argue against the classical enhancer-promoter looping model and are consistent with a new class of models that are coming up from many different sources called the Hub model. I have to say something about the nomenclature here before I explain the model. There is a serious schism, as always, between the academic culture in the United States on the East Coast and the West Coast. Although I'm at Princeton, I'm a Berkeley guy. I mean, I was a professor there for 20 years. I was an undergraduate there. I was a postdoc there. So I'm on rental. I'm on loan to Princeton. So I still think West Coast. On the West Coast, we call it the Hub model. This was put forward by Bob Tejan and Xavier Darzac. On the East Coast, Phil Sharp and Rick Young talk about the condensate model. Okay. Even though there's no evidence whatsoever for condensates, they with great certainty call it the condensate model. I will justify the Hub model on grounds that it's intentionally vaguer because we don't know what's going on. So this is strictly a model, but this is just guides us as to how to think about this coordinate bursting. We know that the two-color reporter gene we know where we inserted the genome. We know where the flanking insulators are, and we know that it forms this TAD or loop domain. And this loop domain brings the shared enhancer presumably into proximity with both of the target promoters. We further speculate that this loop domain serves as a trap for the accumulation of multiple RNA polymerase complexes indicated by these blue ovals. And I have to say, this is the transformative finding, not ours, but a transformative finding in the idea. The old model is you recruit one polymerase at a time to an active promoter. But Ibrahim Sissa and Xavier Darzac have very compelling evidence in their live imaging studies that polymerases form transient clusters or condensates, multiple polymerases. So in fact, you're not recruiting a single polymerase to these active promoters. You're recruiting a cluster of polymerases. Do they form through liquid-liquid condensation? I don't know. Is it some other weak molecular association? The point being there's some kind of mass action going on here, and you're getting molecular crowding within the trap, which does not allow the enhancer to get that close to the promoter. This is the basic idea. So according to this Hub model, you accumulate a critical number of polymerases, 10 or 20, and then the activators bound to the enhancer phosphorylate the polymerases. They're released to give the coordinate bursting of the two genes, and then the process is repeated. The nice thing about this Hub model, and at least in its general sense, and the idea of recruiting clusters of polymerase is that it explains the underlying bursting that is absolutely pervasive, not only in Drosophila, but in all systems that have been looked at. So a very important implication of this model, as I said, is that the enhancer does not have to touch the promoter to activate gene expression. It just has to be in proximity. In fact, it may not be able to touch the promoter because of all this molecular crowding. A single polymerase complex has a Stokes radius of 15 nanometers. So you start putting 10 of those or 20 of those at a promoter, and you've got pretty big distances. And I want to know this distance. And as I said, that distance, we believe, is 100 to 200 nanometers. And we got at the distance by studying a process that will upset some of you called transvection. It's a fly thing. The fly people, I know Alex loves this. I mean, those of us in the fly field feel kind of cool when we talk about transvection. It's a secret calling card. It's like, hey, what's up with transvection? Then you do the secret fruit fly handshake, and you know you're cooler. You know you're cooler than the yeast people, or the zebrafish people, or the mouse people. This is a fly bonding thing. Now, transvection was discovered by one of my absolute supreme heroes, Ed Lewis, a remarkable 65 years ago. I have two heroes in life. Ed Lewis, a distant second. Sandy Koufax, the great Jewish left-handed pitcher for the Los Angeles Dodgers, where I grew up. And my father took me to a lot of Sandy Koufax games. And we referred to him quite simply as the left arm of God. Okay? And my father said, this is the greatest Jewish athlete since Samson. Well, that's a long time ago. So he is a supreme hero. And Ed Lewis, he's up there, but not with Sandy Koufax. Okay. So Lewis discovered transvection in the course of his groundbreaking studies on the ultrabithorax locus. And as you probably know, in the course of these studies, he produced these remarkable four-wing flies, I mean, beautiful flies. And this got him a Nobel Prize in 1995, together with Nusslein-Volhart and Wieshaus. Now, transvection, in its simplest terms, is a specialized form of genetic complementation, which is best explained as regulatory sequences on one homologous chromosome, regulating the transcription unit on the other homolog. It could be either positive or negative regulation. It doesn't have to be an enhancer. Even silencers, even repressors are transmitted sometimes in these experiments from one homolog to the other. So we figured this is perfect. If we could set this up in the early fly embryo, we put the enhancer on one homolog, the reporter gene on the other, and now we just have to measure how close the two chromosomes have to come together in order to get activation of the reporter gene. That's the basic idea of the experiment. So the exact experimental design is shown here. And again, this is the work of Bomi Lim and Takashi Fukaya. They made two different reporter genes, one with PP7, stem loops, and the strong snail shadow enhancer located immediately upstream. Okay, this is within a KB reporter. And they inserted this in a particular location, actually a few different locations of the Drosophila genome. Then they made a second allele, MS2, and inserted the same region of the genome, but on the other homologous chromosome. So the only way to get MS2 expression, because it lacks its own enhancer, is through transvection, the transactivation of MS2 by the shared enhancer located on the other homologous chromosome. And of course this depends, and this is what freaks people out, this depends on pairing, pairing of the two alleles of the two homologs in somatic tissues. And it is a curious fact that Drosophila and probably other insects do have extensive pairing of homologs in somatic tissues. We're not talking about the germline, but in somatic tissues. Most of this pairing occurs in Drosophila at later stages of development and in larvae. There's almost no pairing in the early embryo when we can do our very best image analysis. Because that early embryo, all we have to do the experiments I showed you in the live imaging is put the objective in 15 microns from the surface of the egg, and we capture everything. Everything's on the surface. There's no action in the middle. So we can use pretty good, super resolution methods of measurement and imaging at that early stage, but not at later stages. So to get pairing to occur, Takashi forced the issue by placing these gypsy insulator DNAs upstream of MS2 and PP7. And those insulators mediate looping in cis, as I showed in the hub model, but they also mediate looping in trans, or pairing in trans. And this does allow us to start observing activation of the green reporter, MS2, by the shared enhancer. And this is a typical nucleus in a live embryo expressing these two reporter genes inserted in trans on the two homologous chromosomes. Shortly after mitosis, at the early periods of nuclear cycle 14, that final interphase, leading up to gastrulation, you only see red bursts for the first 10 or 15 minutes. And this is because it takes a while for the two homologous chromosomes to pair. If you think about it, at mitosis from the previous cycle, the two homologs have to independently align on the metaphase plate. So they're in different locations of the nucleus in the interphase, initially in the interphase of the next cell cycle. And then it takes a little while for these insulators to bring the two alleles together. Once they come together, we can visualize this directly, you start getting activation of MS2. But to our surprise, and I think there's something kind of elegant about this, whenever we see transvection, transactivation of MS2, it's always accompanied by activation of the cis-linked PP7 gene. So when we have a shared enhancer, we get coordinate activation of the link reporter genes in cis, but even in trans across homologous chromosomes. I think this is pretty strong evidence for some aspect of the hub model. The next slide shows a very beautiful movie. I can say this because it's not our movie. Very beautiful movie of one nucleus like this. It's not exactly this nucleus undergoing coordinate transvection. And this is done by somebody you're thinking of hiring, and I think you should hire him, Mustafa Mir. I mean, I would hire him at Princeton, for sure. This guy actually knows how to build lattice light sheet microscopes. A lot of people bullshit about it. He's the guy who actually builds the scopes. You want that technology here, he's the man, I'm telling you. So it's like he did this, we've been working on this and working on this, and we sent Mustafa our fly, say, hey, Mustafa, can you make us a movie? Like in one afternoon, he made us like our best movie, like by a mile. I mean, this is amazing. So here is a single nucleus, and here you see the nice coordinate coactivation of the linked alleles. Remember, the enhancer's on the red allele. The green allele is activated by the enhancer associated with the red allele. And they never really merge. I mean, they always remain physically separated, and you can see that they're highly coordinated. They really look like they're sharing a common pool of RNA polymerases. I mean, Mustafa gave us like 3 or 4 movies like this, and he literally spent like 1 hour doing it. So for me, it's like, when he sent us this movie, I felt like the monkey in 2001: A Space Odyssey, the monolith. What is the monolith? What is this technology? Okay. So we interpret these results, again, in the context of the hub model. So I showed you with the cis-link reporters that you form the hub in cis through interactions between neighboring insulators. Here we imagine that the hub is formed in trans by the interactions of the insulator DNAs on the 2 different homologous chromosomes. And once they come together, you create this hub, and it's just what I said before. The model is you trap multiple polymerases, you reach a critical number, the activators phosphorylate those polymerases, coordinately release them to give you this nice coordinate activation of the link reporter genes in this time in trans. So a shared enhancer, we always find, gives coordinate activation of link reporter genes in cis and in trans, and that's our very best indirect evidence for the occurrence of these hubs. All right. So now we've got the perfect arrangement that I discussed before to get at this obsessive question of just how close does this enhancer have to get to this promoter to activate the MS2 reporter gene? What is that distance? And this seems like the perfect setup because you have the enhancer here and the MS2 reporter gene there. So a graduate student in the lab, Tyler Heist, has used a variety of super resolution imaging methods to measure this distance. Now remember, we're measuring this distance through surrogates. We're tagging the enhancer with these nascent transcripts. We're looking at the very beginning of a burst when the signal is within the first 1 kb of the transcription start site. So we have about a linkage of 1 kb between the signal we're looking at and the actual location of the enhancer, and about 1 kb between the signal, the nascent RNAs made by this MS2 reporter gene, and the promoter. Okay? So they're not directly labeled, and frankly, there's no good way to directly label the enhancer and promoter to do these studies in living embryos. You can do them in fixed, and we're doing some work in fixed tissues, but in living embryos, you have no choice but to use surrogates, as we are here. So in this series of experiments, Tyler used the fast ARIScan attachment on our Zeiss 880 microscope, which really gives good XY resolution on the order of 25 to 30 nanometers in the X and Y dimensions. And here you see 2 neighboring nuclei in a living embryo. It's a snapshot where the burst is off here, but you're getting the coordinated bursting of the 2 alleles here with the enhancer on the red allele and the MS2 promoter associated with the green nascent transcripts. And so, Tyler looked at a bunch of these living nuclei and measured the center-to-center distance to get at least a rough measure of the distance between an enhancer and a promoter. And the distance is big, 280 nanometers. I mean, that's a huge distance, if this is accurate at all, between an enhancer and a promoter in triggering transcription. Now, there's a large variance, plus or minus 100 nanometers. Okay, 280, and I didn't have the heart to write this here to freak you out, plus or minus 100 nanometers. But that plus or minus 100 nanometers does not seem to be technical or biological noise. What Tyler observed is that there are natural fluctuations in the distance between the co-expressed alleles during a burst. And I'll show you one movie of that. And we're really in the home stretch. I'm almost done. So here's a single nucleus. Here's the co-expression of the 2 alleles. And this movie runs for about 2 minutes. And you'll see going center to center, this is like an accordion. Every 20 seconds or so, there's an oscillation from a far distance to a short distance, far distance to short distance. And on average, that accordion is moving about 100 nanometers. Okay. So if you take all of this into account, you go to the closest distance that the two alleles are during a coordinate bursting event during these oscillations, and you take into account all the, the noise and the uncertainty, and we also measure sometimes at the edges, not just center to center, I think a very conservative estimate for the distance of enhancer and its target promoter is 100 to 200 nanometers. I think that's a conservative estimate. What does that mean physically? If this was linear DNA, which it's not, and if it was completely bound up in nucleosomes, which seems unlikely, that would be a linear distance of about 3 or 4 kilobases. And that's how we think about it, that an enhancer has to come about within a few kilobases of its target promoter to activate expression. I like that number because when you hit 5 kb or more, you start getting that temporal delay I talked about before, depending on looping. But any enhancer that maps within 5 kb of the target promoter is activated as soon as the activators bind the enhancer. The enhancers really do seem to fall into these two classes of immediate response to the activators bound to the enhancer, and a delayed response depending on subsequent looping. And that boundary is at about 5 kb, which I believe is about the distance, in a linear distance, as to what the proximity is of an enhancer for its target promoter. So the penultimate slide here just summarizes our current view of enhancer-promoter communication. And basically, we believe it's a combination of the classical looping and linking models. So the literal looping models, which are present in most of the textbooks, have activators bound with the enhancer coming into direct contact with transcription complexes like polymerase bound at the promoter. So direct, fairly direct interactions in the enhancer and the promoter. And we don't think that's right because of the molecular crowding. There's just much more action in here than would be allowed for this intimate association. So instead of loop forms, there's definitely a looping component. We believe in loops. But the loop brings the enhancer into proximity with the promoter, a few kilobases, not right at the promoter. And then subsequent communication depends on this traditional linking model where people envision some kind of oligomerization of activators bound at the enhancer and the general transcription machinery bound at the promoter. And that these just all kind of oligomerize. In modern terms, we would say they condensate, they form a droplet between the proteins with the low complexity motifs here and the transcription machinery with the low complexity motifs here. They form a droplet and the gene is on. So that's more or less how we're thinking about this problem, looping and linking. And then I'll just end by reminding you of what I tried to say during the past 40 or 45 minutes. In the first part, I talked about timing. We're obsessed with time. And believe you me, as you get older, you get even more obsessed with time, right? And so I figured if we can understand the mechanism of timing, maybe I can set back the clock before it's too late. I don't have much time. And so those two mechanisms I discussed for the exact timing of the appearance of localized stripes and bands of expression in the early embryo were enhancer-enhancer interactions, mostly negative interactions within complex loci like Eve, and the delay in the looping of distal enhancers to the target promoter in the case of Brinker. I showed you evidence for transcriptional bursting and that transport of an RNA packet on the mitotic spindle during mitosis is really a continuation of the transcriptional burst that occurred in the previous cell cycle. I showed you evidence that enhancers control more the frequency rather than the size of bursts. And what I found surprising, that a shared enhancer can activate linked genes in both cis and even in trans during transvection across homologous chromosomes. And we conclude by doing the measurements in the transvection assay that enhancers cannot and do not come into direct contact with their target promoters. Proximity is sufficient, and we believe that's on the order of 100 to 200 nanometers. And so I invoke this combination of looping and linking to explain the communication of enhancers with their target promoters. And so I want to thank the wonderful people in the lab who did the work. Bomi and Takashi were just like this dynamite team. It was a perfect team. A little tension from the gender disparity, a little tension because he's Japanese and she's Korean, but in a really good, but it was really great. But they really were. They were trying to show each other up. And Bomi mostly won, I would have to say. But they were a great team because he was the experimentalist and Bomi is a chemical engineer who did the computational analysis and all the modeling. Tyler Heiss is a graduate student who started a little later, and he did those measurements, the 100, 200 nanometer measurements and the fluctuations I showed you. And sadly for me, he's wrapping up his PhD. And even sadder for me is that these guys left recently to start their own labs at the University of Tokyo and Penn. So now on this project, I'm down to João Raimundo, postdoc. He still looks pretty energetic. I think he's still got a little life left in him. And João's the one who showed the distal delay, the delay in the looping of a distal enhancer to its promoter. And he was the one who identified that transport of the packet of the RNA. He's very interesting because João comes from Enrico Cohn's lab, and his background is in studying floral patterns. And I've had other postdocs with a botany background. And my N is small, and maybe this is wrong, but these guys have very keen pattern recognition skills. And João really does see stuff that the rest of us miss. I mean, like that transport of that little the RNA packet. João saw that. So with that, I'll stop and take any questions you might have. Thank you.